Hello. We'll start our talk about brachiopods by looking at some of the different kinds of brachiopods and sort of how they change through the ages. Can you see the brachiopods in the picture? They're covered in what I think is maybe a red algae. These ones are. Here's a different one over here, covered in some green stuff, and this little orange guy and these little sideways orange guys, all brachiopods. Couple different kinds. So doing very well there. Doing not so well in the rest of the world in general, although you do find them in high abundance in a few places. So there we go. Here is a brachiopod. So think of it not as a shell, because what we find in the fossil record is usually the shell, and you have to imagine the animal that was inside of it. It's a really short worm with kind of an elaborate set of tentacles for feeding. So this is like the mouth, the sort of digestive organs are down here. This is the feeding apparatus. It's these little sort of fingers, little fleshy fingers, and they're covered in little tiny sort of cilia that are whipping along and moving water. And so they're going to pull water toward themselves and filter out all the little particles. Kind of like what a sponge does, except it's a small unit and it's going to build a shell around itself to stay protected. Although there's one kind of related creature called a phoronid that doesn't bother with the shell. Here you can see looking inside the shell with nice polarized light. You can see all these little sort of hairs that are sticking out. These are called setae, and they are for sensing, we think. So here's a little video. Let's see if we can get this to run. Yeah, there you go. YouTube. It works sometimes. So here's a little brachiopod larva and another little brachiopod larva, and they're swimming along, bumping into each other. Now, at a younger stage of the larva, they do usually have eye spots. At this point, they don't really need eyes anymore. They can sense and they can taste with those tentacles and sense motion and touch essentially with the setae. They'll zoom in here in a sec and you'll be able to see the little waving sort of cilia on the end of the, it's called a lophophore, this feeding apparatus. But you can see in these young ones, the shell actually rotates like this because this kind does not have a hinge. The ones that have a hinge, they can only just open and shut like that. But the ones without a hinge are able to rotate. And they have a special set of muscles called adjuster muscles that help them to rotate like that. So let's carry on. You're going to think of a brachiopod as being sort of like this. So imagine this is the front of the Pac-Man. It's like a little Pac-Man, and in some kinds of brachiopods, they're going to have it's like a tail coming out the back, and that's going to be the pedicle. You're thinking of it like this, which even though it's real dumb, it's going to give you the correct orientations. So obviously, the front of a Pac-Man is going to be this side, and that's the back end. And so the pedicle is going to emerge from the posterior. The anterior is going to be where the part that opens up and then food is going to come in. So it's effectively the mouth. Really, it's just the space between the two shells, but that's the correct end of it. Yeah, and then you've kind of got your orientation. Yeah. And when you're holding a brachiopod, and we'll look at 3D models and lots of real shells in the class, hold it up and look at the middle of the back. And so if the middle of the back looks like it's smiling, looks like this, it's probably upside down. Brachiopods almost always frown like that. So you just flip it over and now you got the correct orientation. So this is the top. They usually have what we call the dorsal valve on the top. It's like the equivalent of our back, but they keep it on top. So brachiopods, this is the front of the brachiopod. They're essentially laying face down in the mud, and I'd frown too if I was doing that. So yeah, think frowny Pac-Man. Frowny little Pac-Man with a tail. We have some modern brachiopods that are still around, and people do wonderful studies on those. I'll show you some pictures from those in, I think, the next section of the lecture. But there are also some exceptional fossils. These are from the Silurian of Hertfordshire, and they show a little orthid brachiopod. And this thing coming out the back, that's the pedicle, so you can tell this is the posterior, which means it would have opened like that. And so there's your Pac-Man. And some of them have one concave and one convex valve like this. That's actually pretty common for especially the ones that I like. If you're just looking at the shell, it will have a hole in it, which is where this sort of meaty appendage called a pedicle used to come out. Sometimes the pedicle, like in this case, is for grabbing on to something to hold on for dear life. Sometimes certain modern brachiopods will use it as a tool for digging. It'll be quite muscular. We think there are some early brachiopods that may have basically used it as an anus to defecate out their pedicle. A strange group. We'll look at those in a minute. All right, now let's start on the types of brachiopods. What are you going to see? And really, when we're saying types of brachiopods, we mean types of brachiopod shells, the two valves, because that's the thing that's going to preserve in the fossil record, but also occasionally some inner parts. They have some bony, not really bone, but bone-like inner parts. Can you spot the brachiopods? Yes, these little pink and white guys. There. There's one, there's one. Yeah, doing great. Good job, brachiopods. So, first thing to know about brachiopods is there are a lot of different kinds, and they're really different from each other. Some of them are anyway. So you got a group here, the phoranids, and they may come in between these other two groups of brachiopods, or some studies will put them just outside these two groups. They are a lot like a brachiopod, except that they never grow a shell. And an interesting thing about them is that despite that, they actually have, I'll show you in a minute, several of the genes that you would need in order to make a shell. It's like they may have lost it, the shell making of genes from some of their ancestors, but they've kept a few of them that they use for other purposes. So that's kind of neat. Evolution does that sort of thing from time to time. Yeah. And I don't know why this stopped. Oh yeah, I do know why this stopped. That's fine. Here we go. We'll just leave that there. There's a brachiopod. They're not clams, I agree. Yeah. So some of the earliest branches, so they're related to things like mollusks. And mollusks also do have a shell, at least some of them do. Yeah, the ones you're thinking of probably do. Um, and if you look at the molecular phylogenies, like where they take brachiopod genes and make a family tree out of those with other genes, like copies of the same gene, sometimes the closest branch to brachiopods, sort of in this order, phoranids will either be inside or very close. Bryozoa, which are the little moss animals, yeah, they're not. They're just animals. But they're kind of like colonial brachiopods, but little tiny colonial brachiopods. Those are usually real close. And then a primitive group of mollusks, which are the chitons. They're called the Polyplacophora. They have, it's usually a front and a back shield and 6 little middle shields, and then kind of a furry skirt around there. It is not fur, but you'll see those clinging to rocks if you ever go to Florida. So those seem to be the nearest branches that are outside the brachiopod world, some of the nearest branches. But if we're talking about what's in that group, you got this early branch, which is perhaps one of the oldest, may be the oldest. And a lot of these sort of lower Cambrian, like the first brachiopods, belong in here. These have a calcium phosphate shell. And calcium phosphate is also what our bones are made of. Now, we're not derived from a brachiopod, but it looks like we may have stolen the like bone making gene apparatus from the same source. Or maybe we indirectly stole it from brachiopods, but there were things running around at the same time as brachiopods that were much more closely related to us in terms of all their other features. But yeah, bones, maybe. Huh. They make them very similarly to how we make them. Not identically, but similarly. And the really primitive groups do not have hinge teeth in their shells. Then there's another group that also doesn't have hinge teeth. It's like a little round plate that's can lift up, kind of like that. Brachiopod, they don't have a hinge. They just sort of lift the entire top off, and then they filter water in. And when they're frightened or they don't like something, then they'll snap back shut, just like that. And so these are the Craniata, but they have a calcite shell. So calcium carbonate, like a seashell that you would think of, like a mollusk shell you would find on the beach. Those are calcium carbonate, not calcium phosphate, which is like bones. Yeah, bone minerals. Then you got this other group. So there are craniids still living. Good. There are lingulids that are still living. Great. And down here, there are several of these groups that are still around. Rhynchonellata, pterobrachiopods, and thesidiids are actually kind of weird looking inside. They're neat. Yeah. All right. And so these are all going to have hinged teeth and a shell, and our shellless phoronids are in the middle. So let's go through these groups one at a time so that you've been introduced to them. And what we're going to do in the lab is we're going to get out the collection of hundreds of boxes of brachiopods, and you can go through them and see if you can find members of these groups. We don't have all the groups, but I'm working on it. I'm still collecting. I'm going to get there someday. So at the really primitive end, and we do have these, are acrotretids. And yeah, they look just about like that. They have a little phosphate shell. Lingulids are still around. Acrotretids are, they're pretty numerous in the Cambrian. And then they go downhill, and the last ones that we can find are in the Devonian. Yeah, Discinids. We found a whole bunch of these on the beach in Namibia just a couple of years ago. These are doing fine. Similar sort of shell, that phosphate shell, to what you would get in a lingulid. But they are going to have no hinge and a little lid that sort of lifts up like that. So, yeah, unusual. Then you got your siphonotretids, and we might have one of those in there. It's either 1 or 0. But they're another one of these primitive groups, and Paterinids are another very primitive group that we may or may not have in the collection. I don't remember. If not, I need to get one. So what you can see on these is that they're generally going to have a valve that has a hole in it, and the valve with the hole in it, the hole is where the pedicle would have come out. So even on these super early versions, there is a hole with a pedicle there. And so that's marking one of the valves. And then in this case, the other valve is going to have kind of a bulge on it. Now that's sort of rare. Usually the pedicle is going to come out of the valve that sticks up more and sort of bulges out. That's more common. Usually the pedicle valve is a little bigger than the other valve, which you can call the brachial valve, or instead of pedicle valve, you can call it the ventral valve because it's like the front of it with them laying face down. And this would be the little dorsal valve that would sit on the back. All right. See, that wasn't so bad. Now we do have pictures of some of the earliest growth on these, and you can find that 2 ways. You can get fossils of really young ones. Those are rare because they're not very well mineralized, and so they don't fossilize very well. But you can also just look on an adult, because if this is the adult, then when it was half as big as it was, why, this is all of its earlier shell. They're preserving their earlier growth stages as they grow. So if you want to see what it was like as a little baby, you just look right up near the tips of the shells, because that's where the original hard covering was laid down, and it's still there, even on the adult. It's kind of neat. It's like they keep a little scrapbook of themselves on their body, yeah, at all times. Maybe that would be tiring if it was a human. What you can see here in these different valves, the sort of ventral and dorsal valves, I believe they're in that order, is that particularly on the dorsal valve, on these things, we know that modern brachiopod larvae have 4 little bunches of these setae in their early stage growth. It's like 2 arms and 2 legs, but it's just little bristles, like you're holding a bag of spaghetti noodles that are uncooked out, just, feeling around with that. And so for the fossils, usually that doesn't preserve, but on the modern shells, when they have these four little bunches, or at least the kinds that have those as a prominent growth stage, will have little bulges here. And so these four bulges show you where the back of each of those bunches of bristles would have been. And so you can look on all these things and say, wow, yeah, okay, these are the one valve and the other valve. On the dorsal valve of all of these things, you have evidence that there were four little bunches of bristles, which means they would have had a similar looking embryo. Even though that embryo is lost to time, you can actually reconstruct it to some extent, which is pretty cool. Here is a little brachiopod embryo, and what they're going to do is they're going to start out and they're going to then fold themselves in half, and those two halves are going to be around the part with all the bristles. The bristles are going to become where they do their feeding, and the little two halves of the body that fold around and then start to mineralize themselves. Those are going to give rise to the shell. That's where the shell is going to form. And so, yeah, you call it a protegulum. That's the first little tiny baby shell that a brachiopod makes. And these things are tiny, much smaller than a millimeter, like a couple of microns for the very first one. And they're not very well mineralized at first. But if you get a very well preserved adult, its little protegulum will still be there. It's usually just like a little bump on the end, like a little tiny mound, then the rest of the shell grows out. These things are going to undergo several metamorphoses as they grow, and all the modern ones do as well. And so we can kind of reconstruct that for the ancient brachiopods, which is sort of neat. So here is the reconstructed larval, everything for these brachiopods. So you got your lingulids, like that would be around now. You've got these others. These are the discinids, like that little sort of circle one that we found lots of in Namibia. And here are two of those early reconstructed forms. And you can see the little bunches where the bristles used to be on those. And so they're reconstructing it. And if it looked like that, then the thing before it probably looks like this, which most brachiopods still do to this day. The one real difference here is that whereas most brachiopods will grow a little shell, which remember that PR is for protegulum, means the thing that protects, in Latin, I think. They're going to have their tentacles, they're first sticking out as a blob, and then they fold the blob up to make these little tentacles with the little cilia, hairs, waving around on the ends of them. So all of these are probably developing, and it's probably only at around this point that they actually start to feed for themselves. The earlier stages of development of these look as though they would not have any efficient way to do feeding. And in the modern brachiopods, where you see larvae like this, it's because they have like a maternal yolk, like a little baby chicken uses to develop. And so they're going to use up all of the nutrients from that yolk from their mother brachiopods in order to grow themselves in the early stages until they can fend for themselves. Some of these things, like Lingula, which is still around today, they basically just skip this. They probably originally came from a version that would have done something like this, because so many different brachiopods go through this stage that you can be pretty sure this is actually the original thing. These lingulids have developed so that once they hatch and are in the water column, we think they are basically able to hunt on their own. It's like a super accelerated early development so that they can start squirting around and gathering food almost instantly, whereas the other ones have to wait until they develop a shell and go through a couple more metamorphoses. So there you go. These are brachiopods. These are their little eye spots and their little dark sort of pigment cups. This is like zooming in on it. Here's a lens cell. So there's a cell that actually is going to block all the light from the center. They're going to use, I believe, the same pigments that we use that make our eyes dark, things like melanin. And they're going to have a little cup of melanin things on the backside, so that's going to block light from coming through from the back, because the embryo is basically clear. And if you're clear, light can come in from any direction, and If you just have a light sensing cell, all you can say is like, yeah, the light's on or the light's off. But if you can block off the back and put a little blob in the center, that's going to act like a lens. And so you're only going to get light coming around this that's going to be focused right about here. And so, yeah, they'll be able to see where they're going as babies. And we think they lose these ocelli later as they develop. But yeah, the babies can see. Aren't they adorable? Yeah. Here's a plate, and it's got lots and lots of discynid brachiopods. These are the ones that are like a little tiny Captain America shield that can lift up and down. And here it is with all its little hairs sticking out. These are ready to feed. This one has, looks like a bryozoan growing on top of it, and all kinds of little worm tubes, or maybe those are foraminifera up there beside it. All the ones with numbers on this picture, this is like an interpretation of something else. The G is a gastropod, kind of a snail, but all these other ones with numbers are going to be discynid brachiopods. And so you can see they clamp down onto something, don't really move around this particular group. We think they stay put and sort of cement themselves on. But then they'll just, open their little lids and feed away when they're ready. All right, so we've covered the oldest group, the group with phosphate for a shell. Now let's cover the second group, the craniates and craniids. So here we go. These are the groups. Craniids are the ones that still exist, and here is a lovely looking craniid. They're usually going to cement their dorsal valve, so they're going to be upside down compared to other brachiopods. Most brachiopods put their ventral valve down and dorsal valve is up, and they'll just lift the dorsal valve a little bit to let water in so they can feed. These guys are going to be like that. So the dorsal valve is going to be cemented onto something else that's hard, and they'll just raise up the lid and let water in, pump water in when it's time to feed. There are several other groups here, and so Craniopsids are probably not a real group. It's probably a loose collection of a bunch of early things, things that seem similar, and they're not craniids, but they're probably not all really closely related to each other. Like some of them are probably closer to craniids than they are to each other. Yeah. We've also got our trimorellids, and look at that. So this is our ventral valve. This is our little dorsal valve, and the ventral valve's usually bigger, and it's usually got kind of a nose on the end of it, just like that. When you do certain types of analysis, these are phorinid worms, and phorinids are essentially shell-less brachiopods. So here's their little feeding apparatus up here. They just look like a little worm. They'll usually burrow down into the sediment or it seems like they're coming out of the rocks in a coral reef, but I don't know if they're actually digging their hole in there. But these are relatives of brachiopods. So, and they're the phorinids. All right, so here's a little of the development. So most brachiopods go through a development like this. So their little egg is going to hatch, if that's even the right word for it. And it's going to form up in roughly this shape. It's going to develop lobes, and these lobes are going to go on to develop into different body parts. And then it's going to settle down, and the middle, I believe, is what's going to become the feeding apparatus. And then these two valves are going to fold up around it, sort of like that. Craniids, very similar sort of developmental pattern. And you can see they've drawn it sort of sideways, so you can see the three little bristles, bunches of setae coming out, whereas there's two big bunches coming out here, and maybe there's also some on the back. And then you can see the lingulids, which are just very similar in terms of how they work, but they accelerate the food catching phase. So they kind of skip some of the early typical development for brachiopods. All right, when a brachiopod is making a shell, the first thing it's going to do is lay down a layer of chitin. Chitin, like in an insect, exoskeleton, for example. It's pretty common. This is also how a mollusk shell, like a bivalve or clam, will go about making its shell. It'll lay down this chitin layer. It's kind of like the underlay that you would put under your carpet if you're laying a new carpet. And then the rug that's on top is going to contain a whole bunch of these sort of proteins, things like collagen, which are going to be found as like connective tissue and stuff that's sort of surrounding cells, extracellular matrix, stuff like that. So they're going to lay down these proteins. They're going to do a little patterning. And then on top of that, there are going to be several different proteins that are going to capture the atoms and molecules that the brachiopod is going to use to make its shell. So they'll capture calcium and phosphate for the calcium phosphate shells and calcium carbonate for the calcium carbonate shells, which are more common. But interesting thing, I think, is that so the red ones, these are only found in Lingula. This is the reconstruction based on the Lingula genome, and Lingula is one of the early ones, the one that develops really fast, very early on. Yeah, precocious little brachiopod. All the ones in green are going to be conserved. You're going to find them as the proteins that make a mollusk shell like a clam, and that make a brachiopod shell, and that make bones in things like people and dogs and cats and whatever. Yeah, you know about bones. So these things are all conserved, and they are used in roughly the same way to do roughly the same thing, to lay down a hard mineral layer so that we have some strength. Now they can be laying it down on the inside, they can be laying it down on the outside. You know, those are differences, and they can lay it down in different shapes. That's kind of what makes us different, but we're kind of trying to do the same thing. Everything in yellow is going to be conserved between mollusk shells and brachiopod shells, or at least Lingula shells. Everything in blue is found in both bone and Lingula shells, but not found in mollusk shells. And there are several of these others that they may be present in all of these cases. We know about like the human version of these, but we're not sure. Like the brachiopod version is different enough that we may be missing it. Yeah. So yeah, really surprisingly conserved. Brachiopod shells are made just like our bones are made, kinda. Yeah, yeah, which is very neat. Now here's what I was talking about before. So at the top, these are the names of some of the genes and like bone metamorphic protein. You don't need to know the names of these things, but know that these are all involved in making shells and or bones. And so here we've got a bunch of brachiopods. And so you can see that in several of these brachiopods, these things are present. So if it's yellow, it's present, and there is more than one copy in the genome. So it's probably fairly important. And there are different slight flavors of it. If it's white, it's present. If it's black, it is missing. And so you can see that phoranids actually have, so these are our phoranids up here. And these are your bryozoa in here. They actually have a lot of the same genes that are used by brachiopods to make their shells. But bryozoans have a bryozoan shell, and sometimes it's not even mineralized. And phoranids have no shell whatsoever, and they're all fine. So they've inherited the same genes, but they're using them for different purposes. Because like a gene makes a protein's going to stick to another thing. It doesn't have to stick to another thing as the first step of laying down the layer of bony material that's going to become a shell. It can just stick to the other thing, and that can have some other function. That's kind of how genes work. It's not the big picture. It is very, very small picture, and you got to kind of build up the big picture in lots of steps. Okay, about halfway done, even though it doesn't seem like it. We've done the first two groups, which are very interesting groups. Now we're going to get to the ones that are more similar to most of the brachiopods that are in our collection. Because these are the ones that did really well for a long time and were absolutely dominant in the ecosystem in the Paleozoic. So let's look at some of these. Now, one of these groups, so most of these, so paterinids and siphonotrettids and lingulats and acrotretids, these are all phosphate shell lingula-like or lingula-affiliated things. So they're all at the top of the tree. They don't even show any craniids in here, and that's fine. Craniids are weird. But this one down here, Billingselida, is a group, and those are the ones that are going to, we think, go on to sort of found this major lineage of brachiopods, the ones that end up being dominant for a while. And you can see from this that they also have the 4 little pouches, so they probably had 4 little tufts of setae in there. That's just the standard brachiopod way of doing business, at least in the early ones. So even though these are placed as sort of totally different branches, really far apart in the tree, they're probably not all that different to each other. It takes a while for these forms to sort of diverge enough where you can really, really tell the difference. And even when they do diverge, the early growth stages are what tends to get retained. That's why baby humans look like baby chickens and snakes and fish, because we're all related, but we sort of are related up to a certain point, then we go our own way. Yeah, that's kind of how development works. Here's some of my favorite brachiopods, and you can see from these things that typically they're going to have a ventral valve, and it's going to have a little groove in it. So here's the ventral valve up here, and they're going to have a dorsal valve, and it's going to have a little ridge on it, which we call a fold, and then 2 other sort of little folds here, and these sort of grooves that you can see there, there, and there, we call those a sulcus. Each one is, so those are sulci. And you see basically the same shape. If it's a dorsal valve, it's probably going to have either a short or a long sort of bulge like that sticking up on it, because that's what happens. If it's a ventral valve, it's probably going to have some kind of little groove on it. Oh, this one, look. See that little tiny mound there? That's the protegulum. That's Brachiopod's very first little shell. It's left right up there, and then the shell grew around it. It's kind of adorable when you think about it. It's like a pair of baby shoes if you still carried them around in your pocket. Okay. And so, if you're looking in terms of development of this group, your craniids are right over here. They're going to have this calcite shell, and look at that. It's very similar development. So they're going to go through either a 2-lobed or a 3-lobed intermediate, and the lobes are interesting because this bottom lobe that's kind of crosshatched like that, that's going to become the pedicle. And so you can see that's there from a very early stage. They've gone to go through a metamorphosis to get from this to that. The top lobe up here, there we go, that is going to go on to develop and become the shell. That's going to fold up. So pedicle at one end, shell at the other end. The stuff in the middle is going to be the feeding apparatus. And you can see in the very early stages, lingulats do go through a very brief, but even look, at that point, they're still mostly middle lobe. And middle lobe is mouth lobe. These guys are going to grow their pedicle later. They'll just, yeah, they don't need it right now. They'll get it later, thank you. Because they start out as little swimming larvae. They're swimming around in the water. And then after that, they will then settle down and start to dig and sort of hide from the tide and then come back up when it's time to feed. But yeah, you see that craniids don't really work like any of these other ones, but they have a similar path of their embryos. You see the one thing they're missing is a pedicle. It's not even there in their younger stages. And they have no pedicle because they're going to cement their little dorsal valves right down, and they don't have a pedicle at any stage in their development. So based on this, you could say, oh, wow, okay, the pedicle may have multiple origins, because it looks like the pedicle is not there on the embryo. There's not even a lobe. And then after we build the feeding apparatus, we're going to grow a little pedicle off the end of it. It comes later. And so that pedicle of something like a lingulate or a disconid may not be the same developmentally as the pedicle of most other brachiopods. And there are groups that just never go through a pedicle phase. They work it out without a pedicle there. Yeah, so kind of interesting that you can get to something that we call a brachiopod via kind of different paths. which suggests that the different groups of brachiopods may not be that close to each other. There may be a lot of stuff that we are missing that would have filled in the gaps, and these are just the ones that have come down through the fossil record and through things that still exist that we can recognize. But if there were a lot of different things like phoranids that are soft bodied in there, I don't know how we would know those were there. It is very, very rare that those soft bodied organisms get preserved. And really, it only happens at rare places and times. It's just like a lottery ticket win, something like that. So if they happen to be in the spot where the weird fossilization was happening, then you may get them. And if they were anywhere else, if they were 5 feet to the left, you will never see them. And because they leave no hard parts, you'll never know they were there. All right, here's our little brachiopod. And so this is the middle lobe on there. Here's our pedicle lobe. And so that's going to end up sort of sticking down in some of these things. And the what was the top lobe is then going to fold up around. This is the mouth right here. It's kind of at the base of this sort of food collecting lobe. The food collecting lobe is going to become the lophophore. That's this sort of tentacle thing, symmetrical tentacled feeding apparatus. And the point of this is to grab food and then shove it into the mouth. That's how they work. Yeah. All right. Next group. And we're now down to the last sort of group. Now, these are the weird ones from the last group. So opalelids, I don't think I have those, or Naucatides, I don't believe I have any of those yet. But maybe by the time you see this, I will have found someone willing to sell me one, because I'd buy one. It's just... There's not a big market for obscure brachiopod types. That's all. Yeah, I've looked. Chiliids, dictyonellids are the strangest looking things. You'll find lots of fragments of them when we go out collecting. Very rare to find a whole one. I've never found it, but I've seen other people who have. Usually not at the sites where we go. And Coutorganids. Coutorganids are the ones that have something like amorphous kind of poking out there where the pedicle should be. And it's probably like sort of a fecal pellet. They're sort of extruding feces through the hole where their developmental pedicle used to be. So, yeah, that's neat. But these are mostly going to have like a little gap in there for something like a pedicle to come out. On this one, it's up near the top. And this one is right between the shells. On this one, they have a kind of a little depression or maybe a hole coming out right there. And these guys have this sort of triangular landing sort of strip on the top, and it looks just like that if you ever see one. Very strange. The rest of these will look like, you'll find them fragmented. They'll look like an old LP, like a 45 record, but just little tiny broken pieces of it. Very strange. They got grooves in them. I've never tried to play one. I don't think they would sound good, but they're pretty awesome. So those are the old ones. Those are mostly things that exist in the Cambrian and then go bye-bye after that. Then you got your Billingsellids, and we may actually have one of these. I'm not sure, or we may be missing this and I need to get it. But these are gettable. They've got a little pedicle hole at the top, at least in Billingsella and a couple of its relatives. And you can see you've got covers now for where the holes were, where the old pedicle used to come out. We're going to develop these sort of cone segment-shaped little covers there. So this one's going to be called a pseudodeltidium eventually, and this will be called a chilidium. Or it's going to be similar to the things that are called by those names more properly. Orthotetids get to giant size like that. Not that hard to find in some places. Strophominids, so the stropho part means sort of flat, like a line. So these guys all have a strophic hinge. Strophominids, orthotetids, and productids. So it's like straight across the little hinge line. And that doesn't seem very unusual yet, but most of the other ones that we'll see have a slightly tilted or extremely tilted hinge line. And so they just work slightly differently. These are like a sort of flat door opening up, and those things are sort of pointed when they open. All right. So early strophominids, which are like these guys, So it looks really different from these other ones. But in the earliest ones that you find right down at the bottom of the Cambrian, they will actually have a little sort of cone on the top, and it's got a little hole in the center of it. And that's where their little, this is their like larval protegulum, where they're just learning how to, fish for themselves, just catch food. And they'll have a little hole where their pedicle used to, it had a little pedicle escape hatch, and that will sometimes be preserved on the actual specimen. And it'll be on the ventral valve because it always is. That's where the pedicle comes out. And then the dorsal valve is usually what's going to support these sort of feeding arms, the lophophore. All right, and here's the rest. These are the ones that make up the majority of the collection. These are the ones that were the dominant things. So we will look at these in class, and you're going to learn what some of these are and how to recognize them, because you need to know this. How are you going to get through life without knowing a spirifer from a pterobrachylid? Won't you be embarrassed next time you're at the museum if you don't even know that? I think you will. I think you know this is true. These are Thesidiids. These are the ones that are still around, as are pterobrachylids and rhynchonellids, these little guys. Rhynchonellids have a very pointed sort of hinge. You can see all these hinges are, these are just a little, like orthids, they're just a little bit not quite straight across, just a little bit tilted. Spirifers are pretty close, but not quite straight across. And some of these other ones are very, very tilted. Like look at that hinge line, just like a little arrow. Pentamerids or pentamerids, I'm not sure how people even say that. Really big during the Silurian, not so big during the Devonian. These are just where some of the big muscles would attach that would help them open and shut the shell. But basically, there's a dividing line that goes right down through the ventral valve and essentially cuts the interior of the animal into two very separate parts. They're living the bipolar life and living it strong. Yeah. So they're kind of interesting. There are several of these with sort of spiral apparatus up inside, like athyridids. You can see it's sort of coiling around here. This is a little bony support that would have held the lophophores. They had spiral lophophores. You also got spirals on these atropids, except that the spirals were sort of flat like this. So the little coils like that. Whereas atropids, they're really short coils. Spiriferids and spiriferinids, they're sort of long coils that stretch out like that. And you can see in the center of most of them, there's going to be a little hole, and that'll tell you that that is the ventral valve. It's always the ventral valve that has the hole, and that's where the pedicle would come out. Some other, like look at this one, it's very sharp triangular hole. So that's a thing you can find. In most of these cases, the larger valve is the ventral valve, and is the one where the pedicle came out with the hole in it. The weirdos are atropids. They're kind of flipped. So this is their dorsal valve on the top, which is big and curved. That's usually not the case. It's usually sort of like that, little and inside out. And they have a little tiny hole at the front, I believe, where the pedicle would have come out. Or at least that is the pedicle valve. It sticks out a little more, which is a thing that pedicle valves do. Like here on this one, that's going to be your ventral or pedicle valve. Same thing with that, same thing with that. So, same thing with this one on the bottom, which is where that giant triangular opening is. Have we come to the end of brachiopods? No. We've come to the end of this section of brachiopods for sure. So see you back for the next one. These things are great, and we have a ton of them. They are what Paleozoic paleontology is made of and runs on. For me, anyway. Thanks a lot.